Light reaction, as the name suggests, that it is a process which is light dependent. This first step is a light dependent process. What exactly happens in this light reaction is solar energy will get converted into chemical energy. Chemical energy in the form of ATP, and a reducing power will also be synthesized. We will name these substances, which substance is known as reducing power. So what is exactly happening is, it is light dependent, so there is something which is going to absorb this sunlight or solar energy, and that energy will be converted into chemical energy in the form of ATP, plus some reducing power will be synthesized. Light reaction, it takes place in grana. So grana, that is those stacks of thylakoid, that is the place where this reaction will take place. The reason for this particular site is chlorophyll pigment, because the membrane has chlorophyll pigment, and that is the pigment which is going to absorb this solar energy. So it is light dependent, chemical energy will be synthesized, reducing power will be obtained, and the site is grana. What exactly is synthesis of ATP? Actually, when ATP is formed, to ADP, that is adenosine diphosphate, one more inorganic phosphate gets added. So it is addition of phosphorus, it is known as phosphorylation, and it is under the control of energy, solar energy. So it is known as photophosphorylation. So this light reaction is also known as photophosphorylation. Photo is because it is light dependent, and phosphorylation is addition of phosphorus. Addition of phosphorus is to ADP, so that it forms ATP. This photophosphorylation takes place in two pathways. One is cyclic and other is non-cyclic. So it is called non-cyclic photophosphorylation, and other is known as cyclic photophosphorylation. The reason I have written non-cyclic first is because it is predominantly non-cyclic which takes place. So if we have to just divide percent wise, about 80 to 90% times it is non-cyclic, and about 10 to 20 times it is cyclic. As the name tells us, non-cyclic means it is not going to end at the same place where it starts. That is why non-cyclic. And cyclic is, it is going to end at the same place from where it starts. So let us take the non-cyclic photophosphorylation first. Before we get into the details of it, let us first very simply understand where is this happening, and then we will draw a detailed structure. This is the thylakoid membrane. We have drawn this structure. So this is the membrane part, and this gap which we see here is actually the lumen. Lumen means there is a membrane here and there is a membrane here also, and the gap between the two is called the lumen. In this membrane is present PS2. Now we know what PS2 is. It's a collection of pigments where there is a reaction center chlorophyll, there are accessory pigments, and there is an electron acceptor also. Plus, here in this membrane also there are some proteins which are electron acceptors. Then there is PS1. Again, what is there in PS1? There are these pigments like accessory pigments, reaction center chlorophyll, and its own primary electron acceptor. So, all these things are in the membrane. In this particular part, photolysis will take place. We will take up this when we come to the details of it. So, this is just to give you an idea where is what present. So, when we talk about the detailed things, we will add up certain things here also. Now, when we make this complete non-cyclic photophosphorylation, in non-cyclic photophosphorylation, both the photosystems participate. So, it starts with PS2. Let us make these pigments here, and we will make one major, which is our reaction center chlorophyll. Here it is chlorophyll A, and here we will write PS2. PS2 means the pigment absorbs 680 nanometers. So, it is written as P680. Even if this nanometer is not written, it is understood. So, PS2. Let us draw the electron acceptor here. So, this is the primary electron acceptor. We are using short form so that everything can be clearly drawn. And this is our sun, from where the solar energy is going to fall on this complex. So, this solar energy which comes, it comes in the form of photons. So, photons fall here. On which pigment are these photons falling, or from which pigments will absorb it? Each pigment will absorb it. Each pigment has the capacity to absorb a particular wavelength. The light which is coming is the white light, which has all various wavelengths in it. So, some pigments will absorb around 300, 400 nanometers, some will absorb around 400, 500, some 600, so on. So, every pigment is going to absorb according to its absorption capacity. But the main chlorophyll absorbs 680 nanometers. Now, what are these other pigments going to do? These pigments will absorb particular wavelengths, and they will pass on that energy to this chlorophyll. So, all pigments will absorb according to their capacity and will finally pass on that energy to the reaction center chlorophyll. So, reaction center chlorophyll is absorbing 680, but that only that much of photon or that much of energy is not enough. So, all these pigments will absorb different wavelengths, and the complete absorbed energy will be passed on to this chlorophyll A. So, why we call these other pigments as accessory pigments or helping pigments? Because they are helping this main chlorophyll get sufficient energy so that the reaction can start now. So, these accessory pigments absorb various wavelengths and pass it on to the reaction center chlorophyll. Now, when the reaction center chlorophyll gets sufficient energy, then a pair of electron gets excited a pair of electrons from chlorophyll gets excited. We just now saw in fluorescence and phosphorescence how it gets excited. In that case, it would come back from the excited level to the ground level, but here when it gets excited, it is accepted by a protein or an electron acceptor, the primary electron acceptor. Now, the electron has gone to primary electron acceptor. From this primary, okay, this is PS2, so this primary electron acceptor is going to be either colorless chlorophyll A molecule or the quinone, so let us write Q here. Now, this electron which is here is at a higher level. Why is it at higher level? Because these electrons got that extra energy and they jumped from the ground level to the higher level. So, these electrons or this electron pair is at a higher energy level. These electrons then pass through a chain of electron acceptors. See, what we are talking about is, suppose this is PS1, we said this is the chlorophyll here, electron jumps, here we made the electron acceptor, and now the electrons are going to come down the gradient. They will pass through the chain of electron acceptors. So, what we have shown here in details is what exactly is in the membrane only. From here, the pair of electron jumps, it is accepted by the primary electron acceptor, that is Q in case of PS2, and now it will pass through a chain of electron acceptors. There are 3 electron acceptors. The first one after primary, the first one is PQ. Somewhere here let us write down what exactly these abbreviations stand for. PQ is plastoquinone. Plastoquinone. Then here the next electron acceptor is a cytochrome complex. So, we are writing CY only, but it is a complex, it has multiple cytochromes. So, it is cytochrome complex. And PC is plastocyanin. So, third electron acceptor is plastocyanin. So, there are 3 after the primary electron acceptor. Now, what happens? Okay, let us draw it in a same color, that these electrons are now traveling from the primary electron acceptor to plastoquinone, the next electron acceptor. Now, when they move from plastoquinone to cytochrome, some energy, that extra energy which they got, is released. So, here that energy is given out, and this energy will be used to synthesize ATP. ATP synthesis does not take place here, that we will see later on when we come to chemiosmotic theory. So, the electrons when they move from, and let us keep writing this pair of electrons which is moving. So, when they move from primary electron acceptor to plastoquinone, energy is not given out. When they move from plastoquinone to cytochrome complex, all that extra energy is released. That energy is used to synthesize ATP. Let us remember that ATP synthesis does not take place here, but because we are writing the cycle and we have to show what exactly is happening here, we will write that here ATP synthesis will take place. How is this ATP synthesized? To make it even simpler, we can even write it like this: ADP using this energy to ATP. So, this becomes little more clear. So, what are we doing? We are using this energy, or rather the plant is using this energy to convert ADP into ATP, phosphorylation, addition of phosphorus. So ADP combines with inorganic phosphate, which is written as Pi. Using this energy, ATP will be synthesized. So that is in accordance to the name, phosphorylation. And this energy which is coming here, actually from where is it coming? It is this original energy. So it is solar energy which was taken by the electrons. They got excited, they were accepted by the acceptor, electron acceptor, because of their energy level. Then because of this higher energy, they kept moving from one acceptor to the other. At one place they give out that energy. Now that energy is used to synthesize ATP. ATP is the form of our chemical energy. So solar energy has been converted into chemical energy, light dependent, so photo, addition of phosphorus, so phosphorylation. Now we will see why non-cyclic. So photophosphorylation is clear. Now this electron pair has released all that extra energy and comes down to the third electron acceptor, that is plastocyanin. From plastocyanin, this electron pair will go to another photosystem. Let us come back to this one. What did we start with? We started with PS2. The electrons get excited, go to primary electron acceptor, then come down this chain of electron acceptors, that is, the first one is plastoquinone, the cytochrome complex, and plastocyanin, and then taken by PS1. So from PS2 it has come to PS1. So this is PS1, and we will draw this chlorophyll, which is the reaction center chlorophyll. Again, it is chlorophyll A, but it is a part of PS1. So it absorbs 700 nanometers, so it is written as P700. Now this pair of electron is taken by this chlorophyll. So now electron which was released from the chlorophyll of PS2 has reached the chlorophyll of PS1. Here also there was sunlight which was coming, here also there is sunlight. So on this photosystem also there is sunlight. So here also there is sunlight which is falling on this. What are these accessory pigments going to do? They will absorb and pass on the energy to the reaction center chlorophyll. This reaction center chlorophyll got 2 electrons from PS2. This photosystem also has its primary electron acceptor. In case of PS1, it is either iron sulfur protein or maybe some unknown thing. So let us keep this blank. Now, this pair of electrons again gets excited. Why does it get excited? Because this pair of electron will again absorb photons, plus the accessory pigments will also pass on the energy to the reaction center chlorophyll A. These electrons again get excited, and from the ground state they come to the excited state. Here they are accepted by again an electron acceptor, the primary electron acceptor. From primary electron acceptor, this pair of electrons is accepted by an electron acceptor which is written as FD. So let us write this FD here. FD stands for ferredoxin. So, in case of PS1, from primary electron acceptor, the electron pair goes to ferredoxin, and from ferredoxin, this electron pair is accepted by a substance called NADP. NADP is nicotinamide adenine dinucleotide phosphate. Nicotinamide adenine dinucleotide phosphate. So finally, that electron has gone to NADP. All this is happening here. So now again, let us come back to this original thing and let us see what is happening where. From PS1, the electron was accepted by the electron acceptor, then it went through the electron acceptor chain, it was taken by the chlorophyll of PS1, again it went to primary electron acceptor of PS1, ferredoxin and all these things are here. This NADP is here. It is in the stroma part, and this electron which is coming from this ferredoxin is taken by this NADP. So this electron pair actually goes to NADP. This is part 1, it is not complete yet. Now let us talk about some different thing. This chlorophyll A of PS2, what has happened to it? Because of this energy from the sun plus the energy which is collected and supplied by the accessory pigments, it lost a pair of electrons. Light is going to continuously fall on this chlorophyll, so it will go on losing electrons, and we know if electrons are lost, then the atom is going to change into an ion and the properties will get affected. So there has to be some process by which the lost pair of electrons should be given back to this chlorophyll. That is done by a process called photolysis of water. Water molecule. So this sunlight, it even falls on this water. Where is this water? This water is in the lumen. So here this water is going to split in presence of sunlight. It is known as photolysis, or it is also called photooxidation of water. Photooxidation of water. Water molecule splits into a pair of electrons, a pair of protons and half molecule of oxygen. Actually one atom is released, but because we cannot write single atom, that is a nascent oxygen, we can double it. If we write 2 water molecules, this will become 4 electrons and this will become 4 protons and 1 water molecule. But because we want to just keep a track of these electrons, we are showing splitting of 1 water molecule. Now what happens to these 3 things which are produced? How many electrons did this chlorophyll lose? It lost a pair. So this electron pair is given to this chlorophyll. Its loss is made up. So every time light falls on it, it loses a pair of electrons. Simultaneously, 1 water molecule is going to split and it releases 1 pair of electrons which will be given to this chlorophyll. So it loses 1 pair, it gets 1 pair, so its structure will not get affected. What is about this pair of protons? This pair of proton is given to this NADP. So, the pair of proton goes here. And this NADP changes into NADPH2. This NADPH2 can be written in two ways. Either we can write it like this or NADPH plus NH plus. So, this is a more technical way of representation, but we can simply write it as NADPH2. This NADPH2 is the reducing power. In case of light reaction, we started with saying that ATP will be synthesized. So, ATP is synthesized in this particular part and we will also get a reducing power. So, this reducing power is in the form of NADPH2 plus photolysis of water. Why is photolysis required? So that the lost electron pair can be replaced from the chlorophyll molecule. What about this oxygen? This oxygen is given out as byproduct. So, oxygen which is released as a result of photosynthesis is actually coming from water, and where is it produced? This is produced in the lumen. So, water splits into 2 electrons, 2 protons and oxygen. This oxygen will be given out. Now, this electron is immediately given to this chlorophyll. This proton particularly this one has to go here, it has to go here. It cannot cross the membrane on its own, it will be helped by some other protein molecule. So, here there is going to be one protein molecule, it is known as ATP complex. So, this one is ATP complex. We will talk about this ATP complex when we come to chemiosmotic theory, but the proton will come out only through this, it cannot cross the membrane on its own. So, now this completes our non-cyclic. Why did we call it non-cyclic? Electron was first getting released from chlorophyll A of PS2, then through primary electron acceptor chain it went to PS1, and from PS1 again through electron acceptors it went to NADP. Did it come back to the original chlorophyll A? It did not. So, because it is not coming back to the point from where it was lost, it is non-cyclic. So, now we know why it is known as non-cyclic. The electron which is lost from chlorophyll A of PS2 does not come back to the same chlorophyll. So, that is why non-cyclic. Photophosphorylation, it is all light dependent. So, photo and addition of phosphorus to ADP is taking place to synthesize ATP. So, phosphorylation. So, this complete cycle represents non-cyclic photophosphorylation, and during this process two things are obtained. One is ATP, other is the reducing power. Both these things will be used or utilized in dark reaction. Plus, the byproduct of photosynthesis, that is oxygen, is also given out during this non-cyclic photophosphorylation. Now, let us take cyclic and then we will compare both these, that is non-cyclic with the cyclic photophosphorylation.